Michael was educated at the University of Otago, gaining a PhD in biochemistry, a postdoctoral fellow at the University of Uppsala in Sweden, and Durham University in the UK. A decade of plant breeding with oil crops in Sweden for the chemical industry at Swalof Weibel and BSAF Plant Sciences. He's also been involved with the startup of a small biotech company. Michael spent 2 terms at AgResearch working in the animal genomics, firstly on cattle and then moving on to sheep. After retraining in economics, so that probably means 4 universities, he went to London working in health economics. Then he moved over to Pfizer Animal Genetics, based in the US and Australia, working mainly in the cattle industry. Currently, Michael is working in the Department of Mathematics and Statistics at the University of Otago, and he is a lead scientist to beef and lamb genetics. Michael, who's obviously pretty well qualified, is going to step us through what is single step. Michael.
It's strange, I actually feel nervous talking in front of farmers about this kind of stuff than the mathematicians in the maths and stats department, so I'm not sure what that means. But what I'll do is I'll just give you a little bit of a historic perspective, and then I'll tell you a little bit about what single step is, and then give you an update on the status of it. Okay, so some historic perspective. What's this thing? Oh, okay, so I've got 2. So really, what we've been, as humans, we've been doing about 10 or so thousand years of domestication in plants and animals. And then probably in the 1700s, probably the first real performance recording and introgression and progeny testing, and the first herd book in 1791, and that was in the UK. And then really, it wasn't until about, we had Darwin in 1859 with his Origin of Species, and then we had Mendel in 1865. So really, breeding was long before we understood DNA and inheritance. So it wasn't really until about 1906 that really Mendel's work was fully appreciated. So then a lot of stuff went into theoretical population genetics. This is people like Fisher and Hardeen. And then probably the foundations for what we term quantitative genetics, which is essentially mixing statistics with genetics, and and we come up with things like indexes, the mass that we get the breeding values with, really isn't until the 1930s to the 1960s. Okay, and then really in the era we're at now, the breeding value technology really wasn't used until the 1980s, really, when we had compute infrastructure available where we could actually solve large numbers of equations, etc. And then coinciding with that really is progress in molecular genetics. So this is DNA sequencing and genotyping and all that kind of stuff. And then later on SNP chips and that. So really this is where we are, but it's really just a blip in this whole history of where we are progressing as, in terms of using livestock and plants for production and that. So really practical use of GBLUP, genomic predictions, which is kind of where the sheep industry is at the moment. We're doing genomic predictions, and then we're migrating into single-step GBLUP, which is really the focus of this talk. And then we've had a bunch of gene tests like Myomax, etc. as well. Okay, so this is a quote from a paper 17 years ago now, actually, and a lot of the authors are actually in the audience here. There's Shira Lan, who's just spoke, Ken, who probably should be here, John Clark, who isn't here, Dorian, and John McEwen. So this was 17 years ago, and they basically said, using commercially available hardware and recompiling the programs, we should be able to do about 1.6 million animals in an evaluation. And this system should be able to handle all the requirements in the foreseeable future. Okay, so this was 17 years ago, and I think really we've been using that infrastructure for the last 15 years, and we've revamped that, and it's really only in this last 3 or 4 years that we're having a really good serious look at the software and saying, you know, what do we need to do here? So really, if I was just steering the distance into the future, we'll have many, many millions of animals in an evaluation, and I think Andrew's talk reiterated this, and a massive increase in DNA data. So this includes genotypes, but also it'll be full sequence data as well. And then there'll be also a massive amount of data collection from automated electronic devices. And this is from things like processors and that, and also from commercial farms and that. So the volumes will increase dramatically. Number of traits will probably increase dramatically. There'll be more complex breeding objectives, but it really is going to be about big data. So if we go back to this slide, this is probably the era that we're sitting in at the moment, and it's about volumes of data and lots of it and complexity. So currently, what are we doing? This is the multi-step process that we're doing now for genomics, and you'll know that as Sheep 5K if you do those tests. So we do something what we call a calibration. We drag in any data that's available, and we try and reuse it to get what we call a training population, and we use genotypes, we predict a molecular breeding value, and then we do a whole bunch of other stuff, and we mix the breeding value from a national evaluation, which you'll know as the National Genomic Evaluation, and a bit of horsing around, you know, blending in that gives you a GBV, and that's what you see on your reports. So essentially, the single step, which is I think is a good thing, is actually a simplification. It takes the genotype data, the pedigree data, the phenotype data, and it just does it in one evaluation and gives you the GBV. So it's actually a simplification from my perspective. The good stuff about it, I think, is that, and I think Dorian will cover maybe some of this later in his talk, is the current system uses 3 evaluations and does a combining. It's cumbersome and probably statistically suboptimal as well. The multi-step is really good and built if you don't have all the data. So if someone sells you a molecular breeding value, you can include it in your breeding values with a process called blending. And I think, yeah, the analogy is probably meta-analysis if you want to think in statistical terms. So using results from an evaluation and combining them and the single step, it'll use all the data in one evaluation to give you a breeding value. And it's nice because it uses the same maths as what basically Henderson dreamed up in the 1950s, which is a really nice piece of elegant maths, I think. So our current multi-step system doesn't push the data through to non-genotyped animals. So in the single-step process, it will. So status, okay. So basically, we can show that there's a superiority for the single-step system, just like what you can in the multi-step or the Sheep 5K system. So with just genotypes, it'll generally give you better than parent average. But you know, I'd put a caveat on this. If there's a lot of information on animals, then your genomics is probably not going to help you a whole lot at this stage. So as with multi-step, the advantage really is for traits that are hard to measure, sex-limited, expressed late in life, and lowly heritable. And this generally means that you've got a low accuracy as a parent average breeding value. Okay, so in my mind, the main advantage really is it resolves a lot of the issues that are inherent in the multi-step system, and that's around bias, and if in statistical terms. So, and what I'll do is basically talk you through a little bit of data that, because I think due to the time constraints. So validation. So the accuracies that you'll get from a single step evaluation are probably going to be comparable with what you'll see in Sheep 5K. So what we do is you can pretty much ignore the alpha here, but the take-home message here is that this is facial eczema, so there's about 9 million animals in this evaluation, and I think we've only put in about 5,000 genotyped animals. So for animals that have very little phenotype information, well, no phenotype information, but parent average, the correlation you'll see between what we think is a reasonable breeding value, and so if you just look at it think of it as an accuracy of the genomic breeding value, it's typically probably upwards of 30 or 40%. And what we generally do is do a division with an effective heritability. So generally that accuracy estimate goes up a bit, but it's probably going to be in line with what we see in Sheep 5K. But really, the important point for genomics is that it's really the marginal increase in accuracy, or how much more accuracy am I getting over parent average, because you're going to have the parent average anyway. So this is weaning weight done for Romneys. Okay, and I've just, or Mohammed has just graphed this as percentage realised accuracy increase just over parent average. So, and what we've found is that weaning weight is actually difficult to show an advantage with genomics because you generally have a lot of data for weaning weight. Each animal has inherently a weaning weight record, but we can show that marginally it does more than often outperform parent average, and particularly in those traits that are lowly heritable, etc. So bias, the beauty, the main reason I think for transitioning into single step, and one of the key is to actually eliminate some of the issues that we've seen in the multi-step system, and also to simplify the system. But here's an example of an issue that we fixed a few years ago. So black spots are the genotyped animals, and the red spots are the non-genotyped animals in a given flock. Okay, and this is for, I think, NLB. So on average, the black spots are lower. So if you look at the intersect, so the genomics, just because it's genomics, it's pulled it down. And this is something we fixed essentially by removing some what we call principal components that were used as fixed effects early on in the development of Sheep 5K. So this is fix this issue here. So we've got a nice straight line of where the mean essentially of the breeding values for genotyped and non-genotyped are not on average different. Okay, so, and this is just a graph then of, there's actually 9 million animals graphed in this plot. Okay, so, and there's about 5,000 genotyped animals. I think this is FE. Okay, so we've got the estimated breeding value. This is what you just see with pedigree alone and phenotype. If we throw in 5,000 genotyped animals, this is it there. So if they were all the same, you'd just have a nice straight line here. The blue spots are the genotyped animals, and they move around more. Okay, so the advantage is that genotyped animals change the most, and the assumption is that the increase in accuracy is mostly for genotyped animals. The black spots are non-genotyped animals, and there's about 9 million animals in that black spot. But they do move around a bit, but not as much as the blue spots. But if you dig deeper, okay, these are 3 flocks, and they don't have any genotype information in that flock. So this is kind of good, you know. Basically, you get a nice straight line, and it's telling you that if you don't have any genotyped animals in your flock and you're just doing pedigree and phenotype, your breeding values aren't really going to change a whole lot. I suspect there's a few things like sire fixes here and this parallel line here, and there's a few animals jumping around, but it's really only a small number. So basically, if you genotype animals in your flock, you'll get advantages, and you'll pull data from other flocks, and that's what you see in sheep 5K. But if you're not doing genomics, your flock's probably not going to change a whole lot. Okay, so in terms of the status and progress of our implementation, so we've basically adapted software that's for the sheep industry here, using software extensively used in the dairy industry. And, you know, we don't really want to be at the bleeding edge, I guess, here, so we're tending to take developments in other area industries, notably the dairy industry, which arguably is way ahead, and we're adapting that. Okay, and we're implementing into a high-performance computing infrastructure using cloud computing services via Amazon Web Services. And I think one of the main reasons here is that we don't have to have a fixed cost here. It gives us scalability. We can go up and down as computing requirements need. And, you know, I think most modern compute infrastructures are moving in this direction. Genotype database, you know, Razier's done a great job in developing a genotype database, and we're plumbing that up at the moment, and that'll be plumbed up with the evaluation system. So we're gearing now to demonstrate and validate the infrastructure, and this is using a SNP chip that an enhanced parentage chip developed by the folks at AgResearch. And so we'll be looking at doing, you know, potentially 20, 30,000 animals to validate the whole system, get everything piped up and tested. And what we'll do is we'll run the system in parallel, a single-step system, with the current NZGE analysis, and plan to go live in 2018. And I think that's me. And then I should just give some acknowledgements. So a lot of the numeric people come from the Mathematics and Statistics Department at the University of Otago, and also at AgResearch in Invermay. AWS is increasingly, we're getting help from API Talent for the cloud infrastructure, and software and support from University of Georgia and Natural Resources Institute in Finland. And also I'd like to thank the BLG team for management, and also for a bit of mana and showing a bit of leadership in this space. Okay, thanks.
Look, thanks, Michael. We're pretty lucky, aren't we, in New Zealand? I think we've got a whole lot of clever Kiwis. If they're not out there designing flying yachts, they're actually out there to actually get the sheep industry down on its daggerboards and flying along. We've got time for one quick question for Michael.
Michael, will the single-step evaluation penalise against sheep populations that have no ability to genotype? And I'm thinking minor breeds here.
So in a way, it completely works. It's quite different from sheep 5K in that we don't have a training population. So in principle, what we're wanting to do ultimately is just replace DNA parentage with a SNP chip. And in a way, that within your flock, that would enhance accuracy. So it doesn't actually stop anyone from a minor breed or its own breed from doing genomics. And if anything, it probably helps them to get into this space. And then the graph I did show that, to reiterate that, if you're not doing genomics and you're just doing basically GBLUP, which is pedigree and phenotypes, your flock and your rankings aren't really probably going to change within flock.